Hello everyone. So in the previous classes we are studying about the the genetic material, and we came to know that the DNA should be the genetic material on the basis of experiments conducted by Avery, McCarthy and McLeod, and later on by Hershey and Chase, and they proved that the DNA is the genetic material. The one of the most important property with the DNA is having is that it has a capability of exact replication, that is it can make exact replica. And secondly, the DNA is most stable molecule, unlike protein on heating, and protein when it come in contact with some acid or bases, it loses its structure, whereas the DNA is very stable molecule. Hence the DNA should be the genetic material. When we are having four criteria on the basis of which we are studying about the DNA. The first criteria was it should have the ability of its replication. Of course, DNA has that capability. Second, it should have capability of minor changes sometime, so that can be done because the replication is not exact always. Thirdly, that DNA is stable molecule. It is stable at a high temperature, low temperature, and even in the pH. Okay. And the fourth, that is it should be able to replicate, it should be able to express the genetic properties which shown by the Mendel. So of course it is showing. So DNA is the genetic material, and in this video we are going to study about DNA. The structure of DNA is actually a double helical structure. So, okay. The structure of DNA was given by James Watson and Francis Crick in 1953 on the basis of observations by Maurice Wilkins and Rosalind Franklin done in King's College, London. Okay, so actual study was done by the Wilkins and Franklin, but they could not understand, they could not give the 3D model. So the 3D model was given by Watson and Crick of the DNA based on to the study of Maurice Wilkins and Rosalind Franklin. Okay, now the DNA is a double-stranded molecule, the two strands which you can see here with the purple color. Okay, and the distance between the two strands is 2 nanometer, which is given here in the bottom. Here you can see. Here you can see down. Okay, and the distance between two helix is 3.4. One complete helix runs from this point to this point, and the distance is 3.4 nanometer, and the distance between two helix are 2 nanometer. Okay, uh, one complete helix has 10 steps. Step number 1, 2, 3, 4, 5. Here the sixth one is hidden. 6, 7, 8, 9, 10. So the 10 steps are there, and 3.4 nanometer divided by 10 steps. So the distance between two consecutive steps is 0.34 nanometer. Okay. The steps are made up of purines and pyrimidines. So purines combined with pyrimidine and pyrimidine combined with purines. There are two kinds of purines and two kinds of pyrimidine. Two kinds of purines are adenine and guanine, and two kinds of pyrimidines are cytosine and thymine. However, another kind of the pyrimidine found, but that is found in RNA, and that is uracil in place of thymine. Now, there is a base pairing rule that adenine can only combine with thymine. So this is This is thymine and this is adenine. Adenine and thymine are combined by two hydrogen bonds, whereas cytosine and guanine, they are combined with three hydrogen bonds. The bond found between adenine and thymine is two hydrogen bond, and bond between cytosine and guanine are three hydrogen. The backbone of the DNA is made up of deoxyribose sugar and phosphate. Okay, so they are making the backbone. Okay, and the bond existing between these two are phosphodiester bond. So where you can find phosphodiester bond, like this one is one phosphodiester bond, and this is second of phosphodiester bond. So this bond is formed with the third number carbon. This is third number carbon, and this is the fifth number carbon. Okay, if I make the structure diagram of pentose sugar. Here it is oxygen, first carbon, second carbon, third carbon, fourth carbon, and this is the fifth carbon. So this is the carbon number fifth, which is having H and OH. Here it is H and OH. Here it is H and OH. Here it is OH and H, and here it is H. So the bond is actually formed with this third number here. This one is the third number, and this is the fifth number. So this one is the fifth one. Okay, so I think it is clear to you. It is a little bit hodgepodge because of the lack of space and my handwriting, as you know, as such I write very poor on the board. So this is actually a computer. So it makes all the more different. So phosphodiester bond. This is the third number carbon, one diester bond, and this is the fifth number carbon, second diester bond. So that is why it is called phosphodiester bond. Okay, and the two strands of the DNA are run anti-parallel to one another. Now, what does it mean? See, this is the fifth number of carbon facing upward, and this is the third number of carbon facing downward. So this is 3 to 5. This strand is running from 3 to 5. This is called moiety, 3 to 5 moiety. On the other hand, here it is third carbon, this one, and here it is fifth carbon. So if we see here, this is running 5 to 3. So the moiety is, this is 3 to 5 and this is 5 to 3. So this is how they are called antibody. In simple, you can see here everyone's oxygen is facing, each pentose sugar is oxygen is facing upwards. Okay. On the other hand, in this, if you see all the pentose sugar, the oxygen is facing downward. So they are running anti-parallel to one another. Okay. Now let us looking at the base pairing rule. Adenine can only form bond with thymine, and cytosine can only form with guanine, vice versa. So the Robert, sorry, what Robert? Okay, this is Watson and Crick was so sure that is a by any means we can split apart the DNA and the two strands are separated. The thymine will only form with the new adenine. The adenine type of pyrimidines are there, then it will form bond not with cytosine, not with another thymine, not with any guanine. Adenine will only form bond with thymine, not with cytosine, not with guanine, not with any other adenine even. And the same is true with others. So even if we separate the two strands, then the newly synthesis strand will be exactly of same type what was existing one. So this is how a new strand will form, and the new strand will be looking exactly similar to that of the parent one. Okay, so that's all in this video, and this is my last video in your class. I'll not be taking any more class now. Next topic onwards, new teacher will be taking your class. Hope whatever I taught all these years is of some use to you. Learn something new every day so that you can cope up with the changing world and the world will be yours. I wish all its success and all the good for your future. Thank you, children.